Jamie Adams, and this is my research partner, Maya Rosenfield, and we're undergrads at the University of Washington taking a marine research course at Friday Harbor Labs.
The focus of our research is to measure the concentration of a marine plastic pollutant, bisphenol A or BPA. We are looking at the concentration of BPA in the Salish Sea in nearshore urbanized areas such as Seattle, Tacoma, and Olympia, and non-urbanized locations around the San Juan Islands.
Our research is important because BPA is a known endocrine disruptor in some marine mollusks and benthic fish, but little is known about the actual concentrations in the Salish Sea.
We're looking at the concentration of BPA in mussels, a known filter feeder, as a proxy for the concentrations of BPA in the surrounding waters of the Salish Sea.
So come with us to the labs and we'll show you how we do the assay for BPA.
So after our mussels have been stored at -20, we remove the shell to get the whole mussel tissue mass and to extract the tissue. After extracting the mussel tissue from the shell, we grind that up with a mortar and pestle with acetonitrile, magnesium sulfate, and sodium chloride, and that helps remove the BPA from the mussel tissue. And we load all of that up into a centrifuge tube.
So these centrifuge tubes are then centrifuged for 4 minutes, and then the supernatant is pipetted into a tube with a 0.45 micron filter on the end. The supernatant is filtered, and this final liquid is what is shot through the HPLC UV.
The machine takes 15 minutes to analyze our sample, and then we get a graph in which we can look for our BPA peak.
For this project, we took 3 mussels from each of 6 locations, 3 urbanized sites and 3 non-urbanized sites from San Juan Island, and we looked at the concentrations of BPA in each of those mussels and then averaged per location the average BPA found at each site. And it was interesting because in Tacoma and Friday Harbor Labs dock, we found a significant difference in the concentration of BPA between mussels at each of those sites. This is the final graph that we get after combining all of our data from all 6 sites. The slightly darker bars are urban sites, and these lighter bars, and then this one with no visible data, are non-urbanized sites around Friday Harbor Labs. And what was really interesting, sort of the most significant data that we got, is that Tacoma and Friday Harbor Dock have significant variance between the concentration of BPA in the mussels from those areas, and that is what the asterisk above those 2 sites means. These higher concentrations found in Tacoma could be due to a number of factors, including higher populations, more wastewater effluent entering the waterways, plastics in the water, but more research would need to go into what is causing these differences in BPA concentration between Friday Harbor Labs and Tacoma.
Friday Harbor Labs has really given us the opportunity to work in small groups with professors that have dedicated their whole lives to working in this environment, in the field, in labs, and really share all of their knowledge with us.
Both of us, I think we've learned a lot about what it's like to work in a lab setting, and we've been really fortunate to have a project that I think we're both really passionate about, and we've had a lot of long nights, but overall the results have been worth it. It's been really fun.
Yeah, we've really been able to link our chemistry courses with our biology courses, and then also get a little field experience as well, collecting the mussels. And so we've been able to work with our mentor, who was a chemist, as well as our professors who are ecologists.
The project definitely made us think about what it would be like to be a real working scientist, and I think we both have the potential to do that as a career.
You can get it if you really want. You can get it if you really want. You can get it if you really want, but you must try. Try and try. Try and try. You'll succeed at last.